You. Hi, everyone. I hope it is audible to the last. Yeah, great. Thanks. First of all, I'd like to thank the organizing committee for organizing it so well and also giving me an opportunity to showcase my work. I'm Tanmayee Samantaray and I'm a postdoctoral researcher at Karolinska Institute. I'm currently working with Daniel Ferreira Research Group, and my main research area is to investigate the disease progression using FDG PET data in dementia with Lewy bodies. However, the work which I'm presenting today is that stems from my PhD research, which I did at Indian Institute of Technology, Guwahati, in India. So yes. So to start with very basic, a network is nothing but a collection of the indigo colored circular spherical shapes, which are called as nodes, and the cyan colored lines which connect them called as edges. So here we see these nodes and edges. If these nodes, they indicate different locations in any particular area, and the edges that indicate the number of vehicles passing through the street, then that is called as traffic network. The same if the nodes are we all sitting here as the academicians or the researchers, and the edges that indicate how many number of collaborations do we have, then it's called a research network. And if the same is in a brain where these nodes indicate different brain regions and the edges indicate the association between any particular specific like metric indicative of those nodes, is called a brain network. Now, coming to the question, can a brain network be encoded? So to address this, can be encoded, that means can it be numerically represented? So to address this question, I proposed a novel algorithm called as Unique Brain Network Identification Number, or in short called as UBNN, for encoding brain networks of individual subjects, not for groups, but for individual subjects. So let's consider this sample network or a toy network with just 10 nodes. And any network could be represented in the form of adjacency matrix, which is nothing but populating with ones and zeros, ones indicative of the presence of connections and zero is indicative of the absence of connections. For instance, if you see there is no connection between 5 and 8, and there is a connection between 5 and 6. So here you can see 5 and 8, there was no connection, so 0. And 5 and 6, there is a connection, that means 1. The here the exclusion criteria which I have considered for this study particularly is not considering the directionality. That means 5 to 6 is same as 6 to 5. And also self connections I have excluded. That means 1 to 1. That means the connection of any particular region to itself is the same, and that is intra. So I haven't yet considered for this, and that is why the lower triangular matrix along with the principal diagonal matrix I have considered as zeros. The next step is the order of input. That means I have taken from bottom to top. That means for node 2, it's 1. For node 3, it's 1, 1. So you can see here 1, 1. And for node 4, it's 1, 1, 0. So it's 1, 1, 0. For node 5, it's 1, 1, 0, 1. So it's 1, 1, 0, 1, and likewise. The next step is converting these binary numbers into their equivalent decimals. So 1 is 1, 1, 1 is 3, and likewise. And then, just for simplicity for understanding, the last row, this purple one, and along with the node labels, I have taken to the next slide. And this is the equation we have formulated to replace the values. So after substituting the corresponding decimal values, for example, here des of 2 is the decimal equivalent of node 2. Des of 3 is decimal equivalent of node 3, and likewise. And after substituting the values, we calculate and we get a value. So this is UBNN 10 is this much value. So this subscript also has a role, which I will tell next. So this number is nothing but which is derived from the network. So this is the number which is indicative of the network. And now it's time to discuss that why do we need to encode a network? Let's say any network that obviously contains huge information. And if you ask me to transfer some hundreds of thousands of networks, I need to transfer the networks, the labels of the nodes, the edges, and all the connections. But instead, I can just transfer this number and then we can reconstruct the network path. So this reconstruction thing I'll tell in the future slide. And this algorithm was an accidental finding when I was just going through a chemistry paper, and the researchers, they tried to convert a chemical structure into a numerical form. And our brain network is a more complicated form of those chemical structures, and that's how I thought it could be used. However, the method which was used there could be used only up to 15 nodes, because they could only find for 15 carbon atoms, which is in a chemical structure. But here, this algorithm could be used up to 1024 nodes. Now coming to the reconstruction, that means from that number, we can reconstruct the network. I said here, this subscript has a role, as in the reconstruction step. We start from the 10 indicates the total number of nodes. Now, there is where we start with. We start with the last node. We just take the integer part and subtract it from the previous value. We get the decimal value. We multiply with 2 to the power n minus 1. N is the node number, so it's multiplied with 2 to the power 8. We get a value. We subtract it from this previous value. We get the decimal value. And again, we find the integer part. That is how we proceed. And we get until the last one where it stops. So you could see here, this is the same as the order of the decimal values we obtained here. We then convert it to binary. The figure, I hope you remember, we converted binary to decimal. So here we convert this decimal to binary, and then we construct the network, the adjacency matrix, which is indicative of the network. So this is how we construct adjacency matrix back from the Eubanyan value. So we tested this algorithm on a real data set comprising of healthy individuals and Parkinson patients from an Indian cohort, which is from National Institute of Mental Health and Neurosciences, India. And I also tested the same on PPMI data set. And the demographics of the Indian data set is shown here. So the DICOM images were converted to NIFTY and then was segmented using CAT12, and the gray matter scans were obtained. These gray matter scans were further parcellated based on LPBLONE probabilistic brain atlas into 56 regions. And then we obtained this subject. For each subject, we have the regional values. And then we construct this individual adjacency matrix based on the correlation value. Correlation was nothing but the reciprocal of the square of the differences. And just to show the pipeline, we have individuals, we obtain their MRI, construct the brain network, and apply our Eubanyan approach to get values. Interestingly, it was found that no two individuals shared the same Eubanyan value. That means every individual have a unique ID number. That is why we named it as unique brain network identification number, or Eubanyan. And this is precisely what we set to achieve here. Just to showcase a few advantages is that it could be used up to 1024 nodes, as already said. It's unique for every individual and hence could be used for brain printing or brain fingerprinting, which is called. It also could be used to study brain plasticity and, of course, space minimization during transfer of the networks. I don't know why it appeared like this. I'm sorry. And this work was published in Brain Sciences on the day of my submission of thesis, thankfully. And to show a few limitations is that larger matrices led to a lot of decimal values. So that resulted to higher mathematical calculations. And another factor is that we have in our equation, we have 1 by 2 to the power I, which again gives a smaller value, and it differs between when we use the same algorithm in Excel and in MATLAB. It gives little different value, as in Excel, let's say 0.000, after 15 places, it rounds up to 1, but that doesn't happen in MATLAB because we can again find a lot of decimal values. I would like to thank Kelly et al. From Cornell University and Lund University, who took time to use our algorithm and our code as well. And they proposed three more methods, which was sort of not exact representation of what we wanted, but they commented that they could find related methods as complete when, let's say, this is the value which we actually found, and that is what they named as complete. And then they rounded up to 15 digits. Here you can see the one here, they have rounded up to 2, and that is what they termed as rounded, and truncated when they did not consider the decimal values and only the integer values. So to address their points and to, while clarifying their doubts, we proposed another method called as modified Eubunin, where in that algorithm, we just started from, you could see that the value was 321 point something, which was the decimal value of the last node. But here it was more of a data-driven method where we did not, choose the node only straight from there, but from the data. As in, for example, we wanted healthy controls. So we took, just to explain this, yeah, we found the node with highest degree. For example, we have different nodes, node 3 with 8, node 7 with 8 degree, and node 11 with 8. So these are the nodes we found, and then we found the principal node of an individual, and that we considered as the one with maximally occurring principal node, devised an equation to represent this and found a value, which is again more of the cohort dependent. And we published it in Brain Sciences as a reply. That is it about the algorithms I developed. I'd like to thank my lab members, Ministry of Education for the fellowship, and my institute, and PPM and NIMHANS for the data. Thank you. And the code and algorithm data is all available in GitHub link provided over here. And if anyone is willing to collaborate or to give any ideas or something, discuss more, you can contact me. Thank you.